Welcome back to the video again, and today we are going to discuss about PCR. Now, what does PCR mean? It is polymerase chain reaction which is done in lab. Now, this is a laboratory technique for rapidly producing million to billion of copies of specific segment of DNA, which can then be studied in a greater detail. Now, what happens? The DNA is something which cannot be studied very easily. In order to read and get proper detail about DNA, you need to do some special procedures. Okay, so polymerase is one of them. So let's come into detail about it. Well, PCR is considered to be one of the most developed molecular technique which has a wide range of applications in the field of medicine, and is also used for broad spectrum pathogen detection and evaluation of emerging novel infection. Okay, now I know these are high terms, but all these mean that from PCR through this technique you can identify a lot of diseases, infectious diseases. Okay, coming to it about its application, PCR is used in research lab, as I've told you before only. Now for DNA cloning. Cloning means producing copies. Okay. So what happened here, that DNA sequencing, recombinant and DNA technology, these are all the process where in lab this technique is used. Now it plays a vital role in genetic engineering, and PCR can be used for the diagnosis of various diseases such as sickle cell anemia, malaria, cancer, inherited diseases and other bacterial and infectious diseases. This is the simple thing here to understand is that when you hear about PCR, the first thing that should come in your mind is that it cures diseases. Okay, it is used to identify about diseases. Okay, now through this we can see that through all these whatever we have learned here, that whatever the problem occurs in the DNA, through genetic engineering we can cure it. Okay, so sometimes it's curable, so through PCR it is done. So about its uses, so diagnose certain infectious diseases, identify a genetic change that can cause disease, and also find some amount of cancerous cell that might be missed in other type of tests. So these were the uses. Now how do they work? So about its working principle, we need to take a sample of blood, saliva, mucus or tissue, okay, from where we can extract the DNA. The sample will contain your own DNA, of course, and possibly the DNA of pathogen or cancerous cell. The sample is then put in a special machines. An enzyme called polymerase is added to the sample. This causes the sample to produce copies. And then in the last step, the process of the copying is repeated multiple times. After an hour, billion of copies are made. If virus or pathogen is present, it will indicate on the machine. So this was very easy and hope you understood. Thanks for watching. Bye bye.